Greetings and an excellent evening to everyone present here. I am P. Tejasvi Prasanna from Proteus, the Biology Club of IISER Trivandrum, and I shall be your host today. Proteus is delighted to welcome you all to our first talk in RHETOR 3.0. RHETOR started as a mini symposium with guest speakers worldwide, indulging our audience in various exciting developments in the scientific arena. We are excited to conduct RHETOR with full enthusiasm and at a larger scale this time. The theme of RHETOR 3.0 is etc., a timeline of biological manipulations. We will explore the concept of the hierarchy of the biological organization from molecular biology to astrobiology and leave it open ended for the future of biology to discern upon the concept. The topic for our first talk will be Unravelling the Collective Dynamics of Biological Cells and Interdisciplinary Inquiry. A speaker for the day is Professor Tamal Das, Principal Investigator and Reader at Tata Institute of Fundamental Research, Hyderabad. He is a DBT Wellcome Trust India Alliance Intermediate Fellow and Partner Group Leader of the Max Planck Society in Germany. We cordially welcome you, sir. Professor
Okay.
Ta-
Yeah,
yes.
please go ahead. Yeah, sorry.
I'm sorry. Yeah.
Yeah.
Professor Tamal Das is an integrated BTech MTech degree holder in biochemistry from IIT Kharagpur, where he also obtained a PhD in biotechnology. He worked as a postdoctorate research scientist at Institut du Cancer de Montréal in Canada and Max Planck Institute for Intelligent Systems in Germany. He was the project leader at Max Planck Institute. We are honored to have you here today, sir. Before we begin the talk, I request the audience to kindly keep microphones muted and videos switched off throughout the session. I request everyone to please hold on to your questions until the end of the talk and let's have a fruitful interactive session towards the end. I would like to hand the virtual mic to Professor Tamal Das now. Thank you.
Thanks a lot. Thanks a lot for inviting me to this forum
My
and
pleasure.
to allowing me to present our work and for your enthusiasm
Thank you,
and
sir.
for this nice introduction.
Thank you.
So as you can see that I'm going to talk about the collective dynamics of biological cells, and we are going to approach this problem from a more interdisciplinary point of view, which combines a bit of physics and a bit of biology. So if I have to define the collective dynamics, so the collective dynamics, by what I mean is that it is the behavior or the dynamics of the many cells together, such that they almost behave like single individuals. So in this problem, what we are doing, we are trying to understand the collective dynamics of cells from the point of view of a physicist. Essentially, we are trying to use many techniques to measure or part of the forces at the cell-cell junctions level or the cell ECM at the cell ECM junction. I hope you can see my cursor. Can you?
Yes, sir. It
Okay, all
is.
right. That's perfect. Yeah, so we are trying to understand this process by measuring or perturbing forces at the cell-cell junction or cell ECM junction. And then what we are trying to do is trying to look at approach the problem from a biology point of view, and then looking at the signaling pathways like a conventional biologist would do, and trying to combine these two pictures. So with this interdisciplinary approach, at this moment, the lab is working on two problems. One is collective migration of cells, was what you see on the left. You create a wound and the cells, these are epithelial cells, these cells move together to close the wound. And then we are also working on a problem called cell competition, where we are looking at the competition between the two population of cells, like the wild type cells, wild type epithelial cells, and then and a population which is bearing a mutation in one of the oncogenes. So we are trying to solve, trying to ask the important question related to this topic, collective cell migration and the cell competition. And all of the things that we are doing, these are relevant to wound healing and cancer. And to this topic, collective cell migration and to the cell competition, our approach is kind of fixed. So what we try to do, we try to use the biophysical techniques, and then we try to ask, can we find some sort of governing rules, underlying principle, or the generic guidance principles of this biological collective dynamics. And suppose we discover a general rule or principle, then the question we ask is that what would be the molecular mechanism that decide these rules. So in this process, we look at the in this process we are looking at both collective cell migration and cell competition from a tissue point of view, as well as the intracellular structural point of view, and sometimes from the molecular perspective as well. And to this end, we are using an array of biophysical techniques like the traction force microscopy, with which we measure the forces between the cell and ECM, and then the force inference microscopy, by which we look at uh, the tension at the cell junctions and so, and then coupling that with so these two measurements are done at the cellular and the multicellular level. And like I told you, we are looking at the different length scales. So we also interested at the molecular level information. So for this, we are using something called molecular tensor sensor, where you have a freight probe within the molecule, and looking at whether the freight probe is stretched or relaxed, you know that whether the molecule is under tension or it is relaxed. And on top of that, we are also using optical tweezer, or at least planning to use optical tweezer to apply and perturb forces on cells. And we are coupling this all of these biophysical techniques with the conventional biology technique like the molecular cloning, gene silencing, and immunohistochemistry. Now with this, again coming back to this problem, so like I told you, we are looking at two instances of collective cell dynamics. One is collective cell migration, another is cell competition. So let me first start with the cell competition, and then I'll switch to the collective migration. So the cell competition, and the instance of the example of the cell competition that we are looking at is a competition between the wild type or healthy epithelial cells and epithelial cells bearing mutation in one of the RAS oncogene, which is the HRAS. Okay. And what happens is that if this mutation arises in our epithelial tissues, then supposedly these healthy cells which is surrounding this mutant population. So here the mutant population is marked with a green fluorescent protein, or use the green, and they are surrounded by the wild type cell, which in fluorescence you don't see anything, which is black, but in the DIC image you do see the cells surrounding this mutant population. So what happens is that when these two population compete, then these wild type cells kind of get rid of this mutant population. So what you can see over the time, that this mutant population is getting out of the tissue as such. And this is actually happens in our tissue, and this is how sort of our epithelial tissue keeps the control on whether the mutated cells are arising, and if a mutated cell or population arises, then it tries to get rid of that. Okay. So this is at the very heart of the very basic tumor suppressor mechanism in our body, and it is called the epithelial defense against cancer. Now with this is epithelial defense against cancer. The main questions are at this moment that what do the cells sense? So suppose a mutated population or mutated cell is arising in the middle of an epithelial tissue. Now the question is, how do the other surrounding healthy cells sense that there is a mutation, there is a cell which is not like them? The question is there a mechanical sensing or mechanical cue, or is there a chemical sensing or chemical cue? And the next question is how the tissue microenvironment influence it. So for this, we use a model. So in this model, what is happening is that this green cell that you see, so they carry a mutation and they express this protein, that HRAS B12, which is a constitutively active oncogene, but they don't express all the time. So they are under a tetracycline promoter. So if you mix these two cell population, and but if you do not have tetracycline in the media, then these two cell population will coexist, because the mutated population will not actually express this protein, that HRAS B12 with GFP. But the moment we add tetracycline into the media, so that will trigger the transcription and the translation of this HRAS B12 protein coupled with GFP. So these cells then will behave like a primitive oncogenic cells, and then they will get extruded from the tissue. So this is a system, in vitro system, where you can watch this tumor initiation process sort of happening in under the microscope, and then you can ask a lot of questions, and then you can do all the measurement. And in this, so we are approaching So to all of these questions, that what do the cells sense? Is there a mechanical or chemical cue, and how does microenvironment influence it? So to all of this, we are approaching these questions or the problems from a mechanical point of view. So what we are trying to do is sort of uh, measure or part of forces that are generated by the molecular motors or cytoskeletons and transmitted via cell-cell junction. And we are going to see that how these things affect the process of the cell competition, which is at the heart of epithelial defense against cancer. Now, with this, the first thing that we asked was the role of mechanical cues in this process. And it has been known, and one of the main mechanical cues come from the extracellular matrix, or ECM. And it has been known for, let's say, last 15, 17 years, that as the cancer progresses, the surrounding tissue stiffens. And somehow this stiffening of the tissue then signals to the cancer cell, asking them to proliferate and invade. Okay. So what is known is that somehow the stiffening of the epithelium is related to the pathophysiology of cancer. But what is not known is that suppose you already have a stiffened tissue because of many things, because mostly because of tissue fibrosis. Maybe you had an injury, you repeated injury at a specific site. Maybe somebody has something like some tissue injury, or maybe infection, like the COVID-19 also caused the tissue fibrosis in the lung, right? So all of this will lead to a stiffened tissue. So tissue will not remain in the epithelial tissue will not remain as soft like it should be. And the question is, if you are predisposed with a stiffened tissue by some mechanism, by tissue fibrosis, then the question is whether this cancer checkpoint, this epithelial defense against cancer through via cell competition, whether this will work or not. So this is the question we asked. So to this end, one PhD student, Shilpa, who has mostly worked on this problem, and whatever I'm going to show related to this is mostly her work, is looked at that if he sort of plays this cell competition over a soft ECM, which mimics the healthy epithelium, and then do the same experiment on stiff ECM, which mimics the fibrotic epithelium, then is there a difference in the outcome? So what we see over here is that on the soft ECM, as you can see, these green cells are extruded out of the tissue plane. So they get evicted out of the tissue. But on the stiff ECM, they sort of exist within the tissue for very, very long time, as long as we have observed. Okay? So it shows that the tissue stiffness or the mechanical microenvironment of the tissue has actually a very significant effect on the epithelial defense against cancer. And so what happens is that till some cutoff tissue stiffness, like the 11 kilopascal, the cell competition and the epithelial, this EDSE, or the epithelial defense against cancer mediated eviction of the tumor cells, that works. So the most of the tumor colonies get this mutated colonies get evicted. But if you are going for the higher stiffness, which mimic the fibrotic epithelium, then on more than half of this population, they do not get extruded. They continue to be part of this epithelium and they continue to grow. Not only so, these experiments were done in using the epithelial monolayer, but we later did similar experiment on 3D epithelial glandular epithelium as well, and we see the similar phenotype. So in the softer ECM, these cells, the mutated cell, get extruded out. On the stiffer ECM, they continue to be part of the tissue. Now the question is, so this is sort of like, if you recall that what I was talking about, that first we try to find out some sort of general phenomena, some sort of generally guiding principle or rules, and then we go for the molecular mechanism. So here you have sort of Silper discovered that this happens, that the extracellular stiffened extracellular matrix attenuates this cell competition. Now the question is, what is the molecular mechanism? So for this molecular mechanism, since she knew that in order to get this cell extruded, the cytoskeleton, the cellular cytoskeletal structure has to get rearranged or modified, she looked at a lot of cytoskeletal protein, whether they undergo some changes on soft versus stiff ECM. And what she noticed is that there is an actin cross-linker protein, filamin, and filamin has a very curious localization pattern. So on the soft ECM, filamin is mostly at the cell-cell junction. But on the steep ECM, you don't see this cell-cell junction population anymore. What you see is that the filamin is surrounding the nucleus. And what she has shown later, that this filamin localization matters. Okay, so for the cells to this mutated population to get extruded, you need the filamin at the cell-cell junction. But on the steep ECM, the filamin is not present in the cell-cell junction, but they are sequestered around the nucleus. So this filamin population is not available for the cell competition or the cell extrusion. And that's the reason that the cell competition or the extrusion doesn't work on the steeper ECM. So I'm just keeping the lot of experiment, lot of signaling, deduction of the signaling pathways and molecular mechanism into just one slide, and you are welcome to go through this paper that we published earlier this year. But what happens is that on the softer ECM, you have a high CDC42 activation at the cell-cell junction, and you have low or no perinuclear actin. You have very relaxed link complex which connects the nucleoskeleton with the cytoskeleton, and you have some low level of protein called FAM101B, which actually recruits filamin to the perinuclear region. So in this is ECM, what happens? So the filamin gets attracted to the high CDC42 activation zone, and filamin goes to the cell-cell interface. But on the steeper ECM, you have low CDC42 activation at the cell-cell junction, but you have high FAM101B, you have higher perinuclear actin, and you have tense link complex. So in this case, filamin get attracted to this location, which is the perinuclear region more, and this perinuclear filamin is not available for competition. So let's say if you need 10 filamin molecules for the cell competition, in case of ECM, 9 molecules it becomes sequestered at the cell at the perinuclear region, and they are not available. And this is the molecular mechanism why the cell competition is failing on the steeper ECM. So having shown that the mechanical cues have a role in the determining the outcome of the cell competition. So the next thing that we are asking is that there is a mechanical signature of this cell competition. Can we measure the forces during this process? So to this, we are using a technique called stress microscopy, with which is we can measure the stresses within a epithelial monolayer. And what we observed is that so what you see over here, again you see you see a field of view where these white zones are basically your wild population, and these black zones are your mutated population. Now I overlay that with first of the cell ECM traction forces, the forces that the cells generate on the ECM, and next I sort of overlay this image with the normal stress, the planar stress in this tissue. And in this, even if you don't really understand that what normal stress is, just understand this thing, that if you are looking at a red zone, then we are talking about the positive stress, which is mean tension. And if you are looking at a blue zone, then you are looking at a negative stress, which means compression. So the red zones are under tension here, and the blue zones under the compression. And as you can see, this mutated population right here, or even you can map that this population or this populations, all of them are in the blue zone. So that means they are getting compressed. They are facing some sort of compressive stress. So it is like the wild type cells or the healthy cell somehow compressing them or squeezing them out of the tissue, which is kind of expected. Now the question is, what is the origin of the compressive stress? Because you don't, so if you have a pure epithelial population, either transformed or wild type, you don't see this much of compressive stress. So the question is, what is the origin of compressive stress? So we looked at systematically looked at different factors like the difference in the tissue stress, difference in the junctional tension, and how the homeostatic area between the mutated population and the wild type population varies. The wild type population tend to be very closely packed, and the mutated population tend to be a little flattened out, so they have more area. And then we also looked at the aspect of migration, and the last thing is the elasticity, and I'll talk about it, how do we go about the elasticity. Now the problem is, in biology, most of the time you cannot perturb one of the parameters without touching the other parameter, because many a times one single thing like the actin cytoskeleton or actomyosin contractility is affecting many of this. Okay, so experimentally teasing out all of these factors one by one is not that difficult or not that easy. And in this matter, what you have to do, you have to approach, if you have a good physicist friend, you have to approach him or her. Okay. So in this case, what we did, we collaborated with one of my good friends, Max B, in the Northeastern University in Boston. So Max is a soft matter physicist, and what Max and his student Shantani, what they did, they built a model of the cell competition and a physical model of the cell competition. And then they looked at that if they varied all of these factors, then which factor actually mattered to give rise to this sort of compressive stress. And what they found that there is the elasticity, the collective elasticity is the most determining factor. So if you have a difference in the collective elasticity between two populations, then you are giving rise to this compressive stress. Now the question is, what is collective elasticity? So if I have to explain you very roughly, just imagine that I give you the wild type tissue as a material, and I also give you the normal and the transformed tissue where all the cells are transformed with this RAS-B12 mutation as a separate material. Now with the same force, you try to squeeze the normal tissue, the wild type tissue, and your transformed tissue, and then look at that under the same force how much the normal tissue gets squeezed and how the transformed tissue gets squeezed. And if you see any difference in this, then you are looking at the difference in the collective elasticity. It's just like how you test the elasticity of a rubber band. The same concept you are applying in case of the cell population or the tissue. Now this is all good, but the question is nobody has measured this collective elasticity as such in this, at least in this format. And without measuring it, all we have a theoretical prediction, but we do not have an experimental evidence as such. So this said, one student, Robert, he took this tough challenge. So he came up with a technique, a novel technique called gel compression microscopy. So in gel compression microscopy, what happens is that the cells are grown over these yellow gels as such, and in the middle you have a disc of expandable gel. So this orange gel is expandable, and by changing the pH of this gel, you can make this gel expand and contract. So what happens is that if you change the pH, these gels will all of a sudden inflate, and this inflation will sort of exert forces on the neighboring tissue, which is your either your wild type population or your normal population. And then you look at that how much they are getting squeezed, how much these populations are getting squeezed. And what he observed is that if you squeeze the MDCK wild type population, and then if you squeeze the RAS-B12 population, the RAS-B12 population gets squeezed or displaced much more than the wild type population. So the elastic modulus of the wild type cells is much higher than the RAS-B12 population. Or in the other way, the RAS-B12 population as a whole is much softer than the wild type population. And this measurement, so if you look at the relative compressibility, the RAS-B12 cells has a much higher relative compressibility, almost 3 times, than the wild type population. So this kind of supports the prediction that it is the difference in the mechanical properties, that the how much force a specific population can withstand give rise to, when these populations are mixed together, give rise to this compressive stress, and as a result of that, which population can be squeezed, they are getting squeezed out from the system. So what I'm trying to say is that although there is a lot of mechanical, although there is a biochemical signals are involved, and as we have shown this protein like filamin is involved, and there is CDC42 and all of this, then what I'm trying to say is this mechanical part of this mechanical cues or the mechanical component of the tissue is a very important factor in determining the outcome of the cell competition. So this is our story till date for the cell competition, and we are now asking question that where does this difference in elasticity come from, whether the tissue fluidity play any role, and there are some other theoretical question that we are asking related to that. Now let me switch to the next topic, which is the collective migration, which we have been working for, so at least I have been working for last 10 years, and the group has been working from since I moved to TIFR Hyderabad. Yeah. So here we are looking at collective cell migration of cells. So maybe many of you are familiar with the single cell migration. And maybe many of you have seen this movie. So this was taken in 1915 by David Rogers at Vanderbilt University. And what you see over here, a neutrophil is chasing a bacteria, and eventually it will be able to engulf this bacteria. So this is something, this is sort of the inception of the cell migration studies. And over the time from this video till date, people have looked at what do you need, what are the molecular signaling involved in the cell migration. And they have looked at many, many things. Like in the front, you need to have lamellipodial structure, you need to have activation of small rho GDPases like RAC1, CDC42. At the back, you have more prominent rho activation, and then you have one kind of actin in filaments in the front and another kind of actin filaments at the back, and the picture is fairly complex, but single cell migration is more or less understood. Okay, now imagine when the cells are migrating together, all of these complex processes has to be coordinated between the neighboring cell. Okay, and this is where the non-intuitiveness of this process or the emergent properties of this process emerges. Okay, now, so that's the most fascinating thing, that the single cell migration itself is very complex. There's an enormous molecular signaling going on. For the collective migration, you have that complexity. On top of that, this complex even need to be synchronized among many cells. Now the question is, can we understand this process from a more simpler point of view? Can we simplify this a bit? Maybe we don't need this much of complexity as such. And the inspiration for this comes from the collective movement that we see in the nature. Okay, so many of you probably have seen these movies or videos or images in Discovery, National Geographic, BBC, and all of these channels. So what you are looking at, the flocks of the starling birds, and then fish school and the sheep herd. And here in this scale, the collective movement, we can give a generic definition. We say it is the correlated or coordinated motion of several individuals. Now, in these cases, these are organisms are the individuals. In our case, the cells are the individuals. And in this scenario, we are the beautiful thing about this thing is that in spite of this all of these complex displays, you can explain all of this behavior by very simple local rules. So if the individual follows some simple local rule, somehow they give rise to all this complex behavior, and this is called the emergence. Something which doesn't exist at the single individual level, but something which emerges at the level of the collective. Now this is the inspiration, but we do not work at the organism level. We look at the cellular level, and the system we have is that we create a wound gap and we let the epithelial cells migrate and close this wound gap, and we look at their motion as such. And when you do that, the first thing you notice that the cells are able to sort of coordinate their velocity over a length scale, which is much, much greater than the individual cell size. And this is the definition, textbook definition of the collective movement as such. Now the question is, how do the cells coordinate their movement over a very long range scale as such? So to understand this, the group of Jeff Redberg and us, what we did, we again decided to measure the forces in this sort of migrating monolayer. And what we can do, we can make a stress map of this migrating monolayer. And at each point, we look at what is the principal direction of the stresses. So that is something we can do using this monolayer stress microscopy. And when we did that and then plotted the local velocity vector, then we observe there is a curious relationship between the local velocity vector and this principal direction of the stresses. So as you see, this is their mutual, the omega is the mutual angle between the principal direction of the stress and the local velocity vector. And you see this omega is very, very biased towards 0. So that means these two angles are, these two directions are kind of mutually orienting to each other. So this movement is called plethortaxis, where the collective motion is driven by the cooperative cell-cell normal or pulling forces. Now this is the generic behavior again, and let's say this is the rule that you have discovered. Now the question is, what is the molecular mechanism underlying this rule? So this is where I ran a sort of a molecular screening. I knocked out many proteins and many proteins and knocked them down. And then I look at in what case I lose this stress following behavior. And it turned out there is a molecule called marlin. There is a protein called marlin. So if I knock down marlin, then I saw that this stress following behavior is gone, and this omega kind of got distributed it broadly between 0 to 90 degree. Now, learning to again going into the more signaling mechanism, marlin has a very curious localization pattern. On standing cell, marlin is mostly localized at the cell-cell junction, but in the moving cell, marlin moves to the cytoplasm. And we saw, and what we have shown that there this marlin moving to the one location of the cell to another location, so like from the cell moving from the cell-cell junction to the cytoplasm, triggers a pathway which helps the cell to make this lamellipodious or leg-like structure in the direction of the forces, and this is how they follow the principal direction of the force. Okay, so this is how, again, one example how the physical techniques and the biochemical techniques can be connected together so that you understand the general rule as well as you understand the molecular mechanism. Another fascinating thing that happens during this collective migration of cell is the emergence of leader cell. So as you can see over here, that there is a cell which kind of migrates first, lead the migration. It has been marked with this orange arrow, and it migrates out of this monolayered zone as such. And these leader cells has been seen. I mean, they have been. We know they are important in wound healing, angiogenesis, and metastasis. And the important question is that if you look at the woundage, it's not that all the cell become leader cell. Only 1 out of 6 cells will become the leader cell. So the question is, how do only some cell become the leader cell? So this is where there is a conventional. So when you started this project, maybe back in 2015 or so, the conventional idea was that there is a hierarchical view. So you create the wound, and all of these cells at the wound margin are equally likely to become the leader. But whoever can trigger this signaling mechanism by which they can form this leg-like structure will become the leader cell, and somehow they will inhibit this. But there was no experiment which actually sort of verified this idea as such. So what we did, we sort of thought that, okay, how does the leader cell, how do the leader cells emerge? And let's study it from a more detailed point of view, and again from the force point of view. So this is where then a student, but now she is a faculty at IISER. What Midhavi did, she measured the traction force landscape as well as the monolayer stresses in this migrating epithelium. And what she observed, that before the cells, actually these leader cells showed their nature, their phenotype as such, and formed this lamellipodia, you already see behind this leader cell there are a lot of force activities. So as if this would be follower cells are priming the leader cells in front of them. So if you look at the behind the leader, the traction forces, and behind the non-leader traction forces, you see there is a significant increase in the traction forces just behind the leader, as well as when you look at the monolayer stress, then you see these two peaks just behind the leaders as such. In fact, by different chemical means, you can tune the spacing of these peaks, and as well at the same time you can control the spacing of the leader as such. So this gives you a sort of systematic view. It tells you that the leader cell generation is not a cell autonomous process, rather it's a non-cell autonomous process and controlled by the would-be follower cell. So one implication is that if you kill this leader cell, and she did kill this leader cell with, let's say, with a high intensity laser as such, then this monolayer will select some other layer, other leaders, and again their separation will be specified by the force landscape in this tissue. Okay, so it shows that this process has a system. This is a system driven process, and the leader cell emergence is kind of a non-cell autonomous process. Again, so we have discovered something, a general rule or principle. Question is, what is the molecular mechanism? So this is where Ritu is a postdoc and early career fellow of the Wellcome Trust, DBT Wellcome Trust India Alliance. So she is asking that what is the molecular mechanism by which these leader cells get selected. And Ritu is very much interested in the signaling related to the lysosome, which is one of the intracellular organelles. And what she observed is that before this leader cell emerge, you actually see in this would-be leader cell, there is a peripheral accumulation of the lysosome that you see in our cell line based model, as well as she did some experiment in Drosophila, and we see the same behavior, that the lysosome accumulated at the cell periphery. Question is, and you can, and if you look at the microscopy image or the videos like this, it becomes very clear. So for example, we have two zones marked over here. One is the lysosome deprived zone, where you have the low amount of lysosome. So anyway, so these dots are basically the lysosome, and the green and the green in is basically the actin filament. So what you are looking at, here in the lower panel, you are looking at a lysosome deprived region, and the upper panel you are looking at the lysosome rich region. And as you can see that in the lysosome deprived region, this doesn't sort of protrude out that much, but when you compare, then you see there is a lot of activities going on, and this region is kind of protruding out. So if I compare again, so if I go back here and then I move forward this to here, then you see that it has made a lot of protrusion while this hasn't done much. Okay, so this shows that the lysosome positioning at the cell periphery matters. Now the question is, how do the lysosome get positioned at the cell periphery? So this is where we showed that the leader cell generation is dependent on the forces, and if the lysosome localization at the cell periphery also influences the leader cell generation, can we somehow connect these two processes? So this is where, with the help of another PhD student, Simran, so what RHETOR did, she generated some sort of designer wound as such, where we can we can create a part of wound where we know the forces will get concentrated. So from our previous experiment, we know so that if we have a highly curved region like this, the forces tend to get concentrated over here. And as you can see over here, the lysosomes also get kind of focused or concentrated in this region. And the next question is, how do lysosomes sort of affect this from the process of cell protrusions, or what we call lamellipodia? So I don't want to go to the signaling detail, but it seems for the lamellipodia, you need to activate a small row GTPase called RAC1. And the RAC1 in the cell exists in two populations. One population inactive, which is GTP bound, which is GDP bound, another population active, which is GTP bound. And what lysosomes do, so when they are localizing to the cell periphery, they bind this specifically to the inactive version of the RAC and remove that inactive version of the RAC from the cell periphery. So you have net RAC activation at the cell periphery, which leads to the leader cell generation. So this is, again, we are talking about some observation, generic observation, or the rule from our biophysical measurement, and then we are looking at the molecular signaling. So the last story I'm going to talk about is something related to the cell polarity. So in the cell polarity, is a very crucial things in the epithelial cells. So if you look at the epithelial cell, then the cells have this something called the apicobasal polarity. So the cell nuclei will be located towards the basal surface of the cell, which is facing the extracellular matrix, and many of the organelles will be facing the upper side of the cells, which is the lumen side. Okay, so this is the organization of a stationary or the standing epithelial cell. But the moment you create the wound and the cells start migrating, then somehow these organelles move from the apical location to the, especially the Golgi complex, move from the apical location to the basal location, and this is very important. Okay, so at least, I mean, as early as in 1970, people have shown this movement of the cellular organelles, especially Golgi complex, matters in the developmental context as such. And if you prevent that, then you have the developmental defect. But from 1970 to 2020, in 50 years, we didn't know, is this process active or the passive consequence of the cell flattening. Just because the cells are getting flattened, does the Golgi simply, it doesn't have a space to stay over here, so that's why it moves to the basal location. So if it is active, and then if it is active, then what is the question is, then the question is, what is the underlying molecular mechanism? So this is where a PhD student, Purnati, she decided to study this process in detail. And what she did, she created a cell line where we can track this Golgi movement in the live cell population. And with this model, what she observed, that the Golgi exists in three polarization states. In one is the vertical polarity, which is there in the stationary cells, that the Golgi is on top of the nucleus. And one is the horizontal polarity. When the cells have started migrating, Golgi has moved uh, to the basal part. But in between, Golgi seem to sort of get dispersed equatorially all around the nucleus. And this is something nobody has observed before. Okay. So she wanted to understand whether how this process, this Golgi dispersion, equatorial dispersion during the migration happen, and whether it is consequential for the migration processes. So of course, we know from our conventional cell biology that Golgi is attached to the microtubule. So the first thing you ask whether the microtubule cytoskeletal network is kind of responsible for this. So what Purnati did, she came up with a way by which by a mutant, and with which she could decouple the microtubules from the Golgi from the microtubule. So this AKP450 protein, so it attaches this, links the Golgi to the microtubule, and there is an so she created a mutated version of this protein, so that it blocks the site so that the Golgi cannot interact with the microtubule anymore. So here in the control, you see that Golgi and the microtubule, they are kind of co-localized. But here you see where the Golgi is located, you see kind of void. Okay. But in spite of decoupling microtubule from the Golgi, what we still observe is that the still Golgi shows this perinuclear dispersal. This all around the nucleus, it get dispersed. So it shows that the microtubule is not important for this dispersion process. So next the question is, then what else? And we know during the cell migration, the another cytoskeletal element that gets sort of heavily modified is the actin cytoskeleton. So it asks the same question, whether the actin cytoskeleton and the Golgi association is responsible for this. So again, she took the similar strategy. So she explored many pathways by which Golgi is coupled to the actin cytoskeleton. And when she disrupted this Golgi linkage with the actin cytoskeleton via a protein called MINA, then what happens is that the cell expressing this MINA mutant, which will decouple Golgi and the actin cytoskeleton, do not show this equatorial dispersion, and the Golgi kind of moves en bloc. So it is there is the apical location one moment, and then it moves to the basal location, but it didn't never go, it doesn't get dispersed. So it shows, and if you look at this video, what you observe is that this actin cytoskeleton from the cortical region, they're forming this kind of hook-like structure, and this hook-like structure is sort of contacting the Golgi and sort of tearing the Golgi apart. So with that, she has come up with a new phenomenon called the migration-induced Golgi apparatus remodeling, or MIGER, where she has been able to show that the Golgi interaction with the actin cytoskeleton sort of tears the Golgi complex apart during the collective cell migration in the epithelial cells and create a new dispersed state of the Golgi. Now, one remaining question is that how the Golgi final polarity, either front of the nucleus or the back of the nucleus, is achieved. So this is an ongoing progress, but we do know that the force polarity, just like we measured the force polarity in our lithostaxis story, the similar way we can measure the force polarity here, and we see that the Golgi polarity and the force polarity kind of align to each other. And now the question is, what is the molecular mechanism that connects the force polarity, the Golgi polarity? But this is an ongoing progress. So the concluding remark is that as you can understand, that we are approaching this collective dynamic problem, cell competition and the collective migration, first from a biophysicist point of view. Using the biophysical tools, we are trying to understand the general rules or principles. And once we understand these rules and principles, we are taking the cell biologist approach, and then we are going after the molecular mechanism. In addition to the stories I present, there are some other important stories are being cooked in our group. For example, Simran is trying to understand that how this curvature will change the wound healing pattern using microfabrication technique. Basil, so we are talking about the so even if you are looking at a genetically homogeneous population, but because of the stochastic nature, there is a fluctuations in the force field as well as the biochemical behavior and the metabolism in an epithelium. So Basil is studying that how this heterogeneity appears and whether it matters. And POS is looking at the intracellular heterogeneity from cell to cell, and this intracellular heterogeneity, how does it get influenced by the mechanical cues like the stiffness of the ECM. And a more recently sort of developing story where Shonok and Simran is working, where we are trying to understand another physical factor, that is the tissue geometry, how that affect the process of morphogenesis using some organoid based model using microfluidics and organoids. Okay, so let me thank so all of the work that I presented, so this was done by the members of the collective cell dynamics lab at the TIFR Hyderabad. So we have this many PhD student and then postdoctoral fellows, and we have of course fantastic collaboration with Mohit at IISc, Max, Freedom, Medhavi, Joachim, Yasu, and in-house collaboration with Manish, Anish, Saroj, and Ullas. And we have been very generously funded by TIFR and DAE, India Alliance, Max Planck Society, very recently HFSP Human Frontier Science Program, and then ACRB. Thank you very much. I'm happy to take whatever question you have.
Thank you so much, sir. That was an insightful and delightful talk indeed.
I
Now,
hope I am on time.
yeah, it was perfect. Thank you so much, sir. Now, I would like to invite any questions from the audience. I think you can put it in the chat box or unmute and talk.
So if something is questions are being asked in the chat box, can you, because I cannot see the chat box, so can you just read
Yes,
it
sir, I'll
out?
read it
That
out
will
for
be
you.
fantastic. Okay.
I
Yeah.
think Satish Kurana sir has something to say.
Hi, Tamal, Satish here.
Hi. Hi.
Very nice talk. I really enjoyed your work, and talk was really interesting. So I have a general question for you.
Yeah, please.
You put your talk very nicely. First part was about the lot of things about cells, not migration, and then it comes migration. A lot of push and pull, plus then the cells they start moving, and then ECM plays important role in it. So, what do you think about the process when metastasis starts as the cell is growing and more and more mutations accumulate, and then the cells decide that it decides, or it turns out that it becomes metastatic. So, does the cell change its extracellular matrix, changes the rigidity property of the local niche? So how does it do it? Do you have any thoughts on it? I know that it is not directly probably your interest, but
Yeah, but this is something which has been studied, because when cancer biologists and the biophysicists got together to understand the, let's say, the physics of cancer, the first thing they attacked was metastasis, because that is the process which is sort of the causes the most havoc, right? So there are a lot of studies where they look at that how this matrix metalloproteases would affect the local tissue property as such. So if I give you the generic answer, there are two things happen. Now, what happens, and this may seem paradoxical, but you have to understand that it happens sort of locally get controlled. Okay, so one of the thing happen is that the tumor tissue is generally stiffer than the, or in general, especially what the data we have from some of the adenocarcinomas like the breast cancer and so. So the tumor tissue stiffens at the metastasis progresses. One of the reason is that this the cells release lot of lysyl oxidase, and that cross-links the collagen. So when the collagen gets cross-linked, the tumor tissue stiffness increases. So in fact, there are drugs which are on trial which actually trying to target this pathway. But at the same time, locally, the tissue chewing or softening has to happen, because the cells has to create pore through which it is migrating. And this is a very interesting topic where people are trying to understand that how the growth and how the proliferation and the migration, which may not be coupled, can happen in this sort of dynamic tissue environment.
Yeah. Just a short question, if I'm not bothering. I think
No, please go ahead. Yeah.
Just one thing. I think the experiment that you were doing was gel displacement. I'm not sure. I'm not from the field, so I probably forget that gel displacement experiment where you were doing.
You mean the gel compression microscopy?
Yeah. So there the gel expands vertically or horizontally, or how does it do? I just missed that point.
Yeah. So the way it works is that you have this expandable gel in the middle, and which is surrounded by let me just I think I'm almost there. Yeah, so here,
Yeah, yeah.
so as you see over here, is that yeah, so as you see over here, so there is this orange part is the expandable gel,
Mm.
and the cells are on top of this disc
Yeah,
surrounding this expandable gel. Okay,
yeah.
now what happens is that, so the moment you change the pH, so what we do, we create a barrier between these two gels,
Mm.
and these cells are of course maintained at 7.4, and this gel is maintained at pH 4. And now we change the pH, we open it up and make everything 7.4. The moment we do that, these gels start expanding, and then they will squeeze the surrounding tissue.
Okay.
And what we are looking at that relatively as compared to the wild types population, how much is the mutated population gets squeezed.
Mm.
So what we can do, we can look at the relative compressibility. Okay.
Yeah, yeah, fantastic. Yeah, thank you very much. Thanks. Very interesting.
Sir, we have a question from Pratiksha Tripati. Can I read that out?
Yeah, please do.
It says, Sir, you mentioned that if there is stiffness in the epithelial cells, the cancerous cell could not be extruded.
Hmm.
I wanted to ask, is there any mechanism that can change this stiff cell into its previous soft form so that cell competition can be easy again?
Oh, that's a $1 million question. If we are able to do that, then that has a very big implications in cancer prevention as such. So you are talking about yes. So some people are working on how to revert the fibrosis, because fibrosis is a major problem, and it can happen because of many reasons: hyperimmune activity, injuries, infection, but fibrosis is also related to the high cancer incidences, and we are offering one mechanism by which these two things could be correlated. But how do you all make the cell again and make the tissue again softer? This is something people are working on, but I don't think at this point there is a very definite tool as such. But if one can do that, that will be fantastic.
Any more questions from the audience? I think it's all done, sir. I'd like to end that. Siddharth Kurne, can you just unmute and ask a question?
Yes, I have a question regarding the softness of the tissue.
Mhm.
Yeah, so like if we change the tissue rigidity, once the
Mhm.
cell has started to migrate already,
Mhm.
will that affect the migration of the cells again?
That's a very good suggestion. We haven't tried that. There is a way of doing it. Oh, yeah. So what happens is that there is something called durotaxis. So in the durotaxis, the cells. Just like in the chemotaxis, they follow a chemical gradient. In durotaxis, they will follow a stiffness gradient in the tissue. Okay. So that happens. But if what I think what you were asking is basically that if we dynamically alter the stiffness of the tissue and then see that how the cells migration happened, right? So that I am not aware of that experiment like this has done in a very controlled way. People have looked at the gradient of tissue stiffness and then looked at how the cells migrated both in vitro and as well as very recently in vivo. And that is exactly called durotaxis. But dynamically changing the tissue stiffness as the cells are migrating, that will be an interesting thing to study. I agree.
Okay, thank you, sir.
Sir, we have another question.
Yes.
Can cell migration take place without leader cell formation? I mean, what is the significance of this leader cell?
Oh, that's a very good question. So cell migration, so the leader cell is doing one thing. So leader cell is generating this lamellipodial structures, okay? Now, if you kill the leader cell, what happens is that the cell migration slows down. But to be very honest, it doesn't get vanished. And another thing is that if you you cannot stop all the leader cells, okay? So what we have done best is that with the laser, we have killed the leader cells. And then what happens is that the slow migration slows down for a bit. And then these cells will create another leader cell and the cell migration will proceed. Another thing that happens when you when you try to get rid of leader cell is that if you look at this, let me pause this video a bit and then look at the just follow the follow this track as such. So as you can see, this orange track in the middle, which is tracking one leader cell, and there is other leader cell over here. So at least in leading front, what you see is a very persistent movement. So this is like a line, so it is not wiggling like this. This is not like a Brownian motion as such. So the leader cells in a way contribute a lot to the persistence of the motion. Yeah. So what would happen is that the individual level, the cell velocities may not change a lot. In fact, without the leader cell, they will try to move everywhere and they will be migrating very freely, but there won't be any persistence in the motion. So this is where leader cell make a big difference. I hope I'm clear.
The same person has another question. She
Yes.
says, if there is no leader cell formation, what will be the case?
So if there is no leader cell formation, so this is something so you are talking about like uniform propagation of the uniform propagation of this of the wound interface without creating a leader cell, or let's say all of the cells generate the all of the cells generate their own lamellipodial structure. So in that case, what happens is that, like I said, that the migration will be faster, but it will not be persistent. It will not be in one direction.
So there's another question.
Yes, please.
Can you please once again describe collective elasticity?
Yeah, so the collective elasticity, what is happening is that so the single cell level elasticity is like if you are taking one cell and then let's say with an atomic force microscope or by some other mechanism, you are pressing that cell and then you are looking at that how this cell is reacting. So that is how the elasticity is done at the single cell level. Now, in the collective cell level, you are doing similar sort of experiment like what we are doing over here. We are squeezing an entire tissue by an expandable gel and then looking at that how much resistance the tissue is able to mount. Okay. So more the resistance tissue is able to mount, or they are more rigid. So that's the idea. But there is a difference between, there is a conceptual difference between the single cell elasticity and the collective cell elasticity. In the collective cell elasticity, the cell interaction matters. So let's say if I have like individual cells not so elastic, but I put them together and then I have these cells put together and then tie them very I mean through the cell junction. If they are connected to each other very strongly, I'll have at the single cell level there won't be any change in the elasticity, but at the collective level in the second case where I will have where the cells are interacting with each other very strongly and they are strongly connected by the cell junction, they will mount more resistance, so they will be more rigid. Okay, is it clear?
Yes, sir. We'll take one last question, sir, and wind up the session. Can I read the last
Yeah,
question?
please go ahead.
Okay. Sir, you mentioned that there is peripheral accumulation of lysosomes due to mechanical forces causing leader cells formation too. But does the lysosomes accumulation change depending on how stiff the ECM is?
This is something we haven't looked at. We haven't looked at that fact. Hello, can you hear me?
Yes, sir. Yes, sir.
Okay.
So, sir, I'd like to end the talk with a thank you note.
Thanks a lot. Thank you again for inviting me. And it has been a very nice forum, and I loved answering all of these very interesting questions. And I
Thank you
wish
so
a
much.
great success to this endeavor.
Thank you
Keep it
so
up.
much. I would like to thank everyone who attended this talk and got actively involved in the interactive session. I extend my gratitude to our faculty advisor, Dr. Sadananda Singh, and the School of Biology for their continued support towards Proteus. Let's meet on 4th November for an exciting talk by Professor Kushagra Bansal. Thank you.